Okay, welcome to the third video in porifera. We left with this generalized diagram of a sponge, and we're now going to move to the formal definitions and the terminology. So the outer surface, panacoderm, we've been through all of this, but if you need it written down, this will probably help. Composed of flattened epithelial cells called panacocytes, interspersed with porocyte cells, also called pores. Those are the holes, the donut-shaped ones. Tubular cells that extend from the external surface to the internal surface of the sponge, and an opening in the interior of the sponge is called the atrium. Right. The internal surface is termed the choanoderm, lined with choanocytes or collar cells that are flagellated. Okay, that big flagellum, that's the whip-like structure. And between the panacoderm and the choanoderm is the mesohyle. That's the jelly-like substance. And the mesohyle consists of a gelatinous, proteinaceous matrix containing skeletal material and amoeboid cells. The mesohyle consists of a gelatinous, proteinaceous matrix, which is the jelly-like matrix, okay, and skeletal material being the spicules. Okay, so spicules. Amoebocytes, another word for them is archaeocytes. They move through the mesohyle, and they function in spicule production, as we said before. So the spicules can either be made of silicon dioxide, which is essentially glass, and these things will have the same light transmitting properties as fiber optic cables. Calcium carbonate is the same thing that shells, most seashells are made from, coral reefs are made from, and protein or spongin fibers. Okay, spongin fibers are spongy. That's the bath sponge type fibers that make up the spongy type feel that you get with a sponge. The compressible, they're very compressible, but they'll spring back. Okay, spicules are mostly found in the mesohyle, but may project through the panacoderm. So if you think about these things projecting through the panacoderm, and if you were to possibly take a sponge and rub certain types of sponges and rub it on your face, you might get a little bit of a rash. You might find that these things, it's very much like fiberglass. These things have little rough projections that can break off. And so if you think about a sponge and what it is, it's essentially a big immobile, living organic mass. Okay, and what's going to happen to something that's big and immobile and full of organic material in the ocean? Well, obviously, what's going to happen is something's going to come along and eat it. So they need to have some defenses, and these spicules, as well as being the girders of a building, being the support structures, they also function in defense. So if we look at this, here are some spicules that have been removed from these different sponges. And if you look through at the sponges, there's a little bit dried out for these micrographs. But what you see is a whole lot of projections that look like palisades from like a Mari Pa, or like sharpened sticks or something like a porcupine. And these things serve as a defense against grazers coming along and just taking a big biteful. And you can imagine if you get a mouthful of things like this and you're chewing up glass, or glass particles, glass shards, that it's going to be fairly unpleasant. So these are one of the defense mechanisms of sponges. So let's have a look at some of the local sponges, and we use these spicules in terms of identifying different species. They can be quite diagnostic. So a lot of sponges may look very similar, but if you look at the spicules, there may be, and the arrangement of the spicules, you might find that they're quite different from species to species. So here you can see this small spiky one here, longer spiky ones in here, and then these other ones projecting through, and they'll be arranged in on all these little small ones through this area. Okay, so this one is a Anchorina alata, which is a gray pillow sponge. Okay, so that, like I said, while the colony shape will not always resemble the one illustrated, the shape and arrangement of the spicules will remain consistent. Okay, this one is the Calispongia ramosa, which is a gray finger sponge, and it's got these very small spicules that are arranged within this lattice, and most of that one is spongin. You'll find that one's quite a bit more compressible than this gray pillow sponge. Okay, Raspalia topsenti, which is a staghorn sponge, usually orange. Here are the arrangement of these boomerang-shaped spicules, and they'll be running in columns out through this body of the sponge. Okay. One of the most recognizable, Tethya aurantium, orange golf ball sponge. Okay, and you can see these little death cookies here that you wouldn't really want to chew up a mouthful of. Little spiky balls, and then they've got these longer spikes as well. Okay, and then you've got the Tethya ingali, pink golf ball sponge. Okay, and very similar to the arrangement of the orange golf ball sponge, but if you look at this, 40 microns, okay, and these ones 20 microns and a lot sharper spikes. All right, so they're a little bit smaller, these little balls, and then the longer spikes as well. Okay, here's this Cleona salata in the spreading mat stage. It's got two different stages that you'll be able to recognize perhaps when you're out diving, but it forms these dense mats. But when it's younger, it just forms little tubules that stick out of the sand. Anyway, so they've got these long scimitar-shaped spicules. It's an interesting one. It's a boring sponge that excavates burrows in calcareous material like shell matter. Calcareous, that's what we refer to when we talk about calcium carbonate, like I was saying before. And they use chemicals to dissolve that calcium carbonate and sort of form a home for themselves. Okay, here's Polymastia fusca, which is a green, really compressible, pillow-like sponge, and it's kind of medium-sized in shallow water, only about 5 meter depth. And if you push down on these, you can push them sort of probably half of their diameter will compress. So they're very compressible. You'll probably see quite a few of these when you're out diving. Okay, and how about this one? This is the Eophon minor. So the bright yellow branching sponge. Okay, and you can see this lattice material with all these very short spicules. So you can use those spicules to diagnose what species you have. Okay, here's a Lucitusa lancifer white flask sponge. Small, brittle, hard, easily crushed. Usually only around 5 to 7 centimeters tall. Quite beautiful, though. Starkly white, and they look fragile and brittle. Polymastia crocus, yellow nipple sponge. Okay, we'll see quite a few of these things. They're very common. Soft and compressible, and they kind of go leathery when they're contracted. And you'll see all the different spicule types here. They're like those scimitar-shaped ones, but not curved that we saw in the other sponge. So, okay. Here's another cross-section of another sponge, of an asconoid sponge. Okay, we haven't talked about asconoid, leuconoid, syconoid body shapes yet, but We will in a second. And you should be able to draw and label this by the end of this module. So here we go, the 3 structural forms of a sponge. So if you're asking, how can sponges get so big if they've only got 2 layers, if they're only 2 cell layers thick with a little bit of mesohyl in the middle? So you've got the outer layer, which is called the what? Right, pinacoderm, and then the inner layer made up of choanocytes, called the choanoderm. So they can't get very big with only those 2 layers, but if they start folding, then what happens is they fold, and they can get bigger and bigger, and so you get water pathways moving through, and all of these will be lined with choanocytes, and then these canals will join up until you get big excurrent pathways. So incurrent and excurrent pathways, when these excurrent openings are called the osculum or oscula. And so we divide them into these different structural forms, but a single sponge may go through all 3 of these forms in its lifetime, and they're found in more than one class, so I'm not too worried that you know these terms. This is the way that sponges can get bigger as they fold in on themselves, okay? And then they form, here's a nice cross-section through a sponge that's quite highly folded in on itself, and you can see all the larger pathways where water has probably already been filtered and is making its way towards the outside of the sponge. We've got the pinacoderm on the outside. Interior won't have any pinacocytes. It's all made up of choanocytes and amoebocytes, and this area is called the spongocoel. Okay, so the spongocoel is the area within a large sponge that is essentially just filtering cells and amoebocytes. Okay, spongocoel. So the interior canals are flagellated or radial canals. Okay, they're flagellated because they're lined up with choanocytes. And here we go. So the seconoid, asconoid, and leuconoid forms again. Okay. So these large sponges, they can filter their own body mass of water every 10 to 20 seconds. So they really pump a lot of water through, and we'll see some interesting videos in class about this. Here's another picture of what looks sort of like a loofah, but it's another bath sponge, or sort of cosmetic bath sponge. But you can see all of the holes going through it, and the pathways where larger volumes of water are grouped together on the way out. And here's another cutaway view of what a leuconoid sponge would look like. So the water comes in, and then you have these flagellated atria, and then the water will exit these collective tubes out to the osculum. Okay, so again, as we've talked about in the last video, the water currents are generated by the beating flagella, and you can pause this and read all this. But they filter particulate organic detritus, and as we said before, they take out a lot of bacteria. So they're very important as environmental scrubbers, as they can take out up to 90% of all the bacteria that pass through their pores. Okay, so we've been through all of this. Phagocytosis we'll illustrate in class. Digestion occurs in the choanocytes and also with the amoebocytes or archaeocytes. And the water flow is used by the sponges for feeding, gas exchange, excretion, and reproduction. So think about it. The water flow, you think about it at first as being a way for the food to come in, but it not only carries in food, but luckily, as these cells create out the products of metabolism, which is essentially like poos and wheeze, out that goes with the water that is flowing out of the sponge. And they also are getting rid of carbon dioxide because they take in oxygen just like we do and respirate, and then carbon dioxide is formed in that respiration. and out goes the carbon dioxide dissolved in the water that's leaving, and also reproduction. So we'll look at that in just a second. But if you're going to have a stationary body form, okay, so you can't move around, you can't really find mates by just sitting there, and so you need to have a way for the gametes, or the reproductive cells, sperm and eggs, to find each other, and that is by drawing in the water sperm have been broadcast into, and then those sperm can fertilize eggs within the body of the sponge. Okay, so these sponges, they have both sexual and asexual reproduction. Okay, they can grow a stolon out from the body. Okay, and here is an illustration of the stolon. These are the little projections, and these things could hit the ground. They could hit the substrate here and start a new colony, or they could break off and then possibly find, if they land in a suitable habitat, then they can start a new colony in that way. Another way is a bud breaking off the parent and drifting away. All right, so which is essentially like the stolon breaking off, and then a piece of sponge being torn off in a storm and lodging elsewhere. So if perhaps, like let's say there was a rock tumbling around in a high energy storm, where a wave moving some of the rocks around the substrate, and it knocks a chunk of this off, it lands right on this sponge and knocks half of it away. If that half lands somewhere else, then it can form a new colony. And in fact, these things are so good at it, that forming new colonies by these pieces breaking away, that you can take a sponge, grate it on a cheese grater in an aquarium, and then because these cells have the ability to become any of the other type of cells in the sponge, then they can reorganize themselves into small sponge colonies. Okay? So sexual reproduction involves the fusion of sperm and eggs, and the eggs and sperm are produced in different parts of the colony. The choanocytes can turn into spermatocytes, so the choanocytes, which are lining the colony of the interior of the cell, can just turn into a spermatocyte. That flagella becomes like the flagella of a sperm, that classic sperm shape with the little tail whipping along, and so they can just break off and start swimming out into the water column when the time is right. If that spermatocyte, there's that word "cyte" again, cell, is lucky enough to be drawn into the body of another sponge, it can be recognized chemically rather than eaten. And if it goes into the wrong type of sponge, then it would just be eaten because it's not the same species. They don't crossbreed. That spermatocyte can be used to fertilize an oocyte, which is a again, as we said, OO, an egg cell. So egg cell. So then the eggs are incubated, brooded inside the body, and then released as what we call a planula larva. Okay, so here we go. There's the sperm coming in, fertilizing the egg. It's brooded for a little while, and then it's a little larva that swims. Little cilia. Again, we see cilia. These things will beat or swim this thing around, carry it in the plankton, and it'll settle and just grow new cells off of itself and become an adult sponge. Okay, so that is very much like that lithotrophic larva that we saw at the life histories and characteristics of the invertebrates. Getting towards the end, we've got the taxonomy, 10,000 species of sponges recorded around the world. So these things, as simple as they are, they all work exactly the same way. They just have the same body structure, but they're still complex enough to have branched into 10,000 different species. All right, and probably quite a few more that haven't been discovered. So you've got Hexactinellida, which are glass sponges. These ones have silicon spicules, and you'll see that these things are arranged somewhat like the X, Y, and Z axis of a graph, of a three-dimensional graph. So they've got right angles. Okay, 6 endpoints, and they often fused, forming a lattice-like skeleton. And they're usually deep water sponges. Okay, so the vast majority of sponges that you're going to see are going to be demospongia. Okay, 95% of all the sponges, and they range from shallow to deep waters. They've got siliceous, so this is silicon dioxide again, and/or spongin fiber, surrounded by mesohyl. Okay, so these are going to be sort of these are the ones that are going to be real compressible. Okay, but most of the big ones that we see in shallow water are going to be demospongia, and they usually are quite brightly colored. And then you have also the calcarea. Okay, and the spicule is composed of calcium carbonate, and exclusively marine. Okay, and this is like your white vase sponge. They look quite brittle, quite delicate. They're very less compressible and more brittle. Okay, mostly found in sheltered positions, rocky overhangs, crevices. Okay. And finally, the class Sclerospongia contains a small number of species found in tunnels and coral reefs. So here's a nice picture of a sponge that has long spicules growing out of it. Alright, now we've said that all sponges work exactly the same way. They just filter out bacteria and the like. And so, as always, there's an exception to the rule. And so we'll have a look at some of these little exceptions. This is the first one. Here's another one. See, that's how small this thing is. Sponge on a stick. Okay. Lollipop sponge. There's another one. Okay, it's got these long projections. Actually 2, 3. These are another type of sponge. Diploma. Okay, and they've got little hooky little bits on them. Okay. How about this one taken from quite deep water? Funky looking sponge with these little structures coming off of it. Two different varieties. They don't glow like this, but they're that's just backlit from the or for the photography. And here's another one as well. Alright, so these ones are some of the spicules. They've got little hooks on them. So what do you think might be happening with these and these? These are actually called carnivorous sponges. So something that has no way of no teeth or no way of sort of biting something or eating a big organism, they still manage to be carnivores. Okay, and so what's happening here? These things will actually, incredibly, these are quite sticky, and these will catch organisms that are moving past. And these ones, you can see these hooked spicules that'll snag things as they're moving past. These things are essentially farming bacteria. Okay, they can grow fibrils out to what they catch, and then ingest these things. But they also are essentially farming bacteria. If something dies on this surface, then there'll be a lot of bacteria that'll be released and would sucked into the interior of the sponge to be ingested. So it's really quite amazing. And generally in biology, just when you think you know something about how something works, there'll be an exception to the rule and surprise you. Okay, let's end.